and I'm here to present you the work that has been recently published in Nature Chemical Biology. And so before I start, I'd like to put that all work in context of the project it belongs to. And so this project involves many people, so the group of Barbara Imperiali at MIT in the United States, the group of Daniel Choquet that I'm working in right now, the Bordeaux Imaging Center, and the group of Laurent Groc. And so the idea behind that project is to use a multidisciplinary approach to achieve two main goals. The first one being to develop tools or methods to be able to achieve acute perturbation of the glutamate receptor synaptic stabilization. And in that respect, there's been many articles over the last few years showing that PDC domain mediated interaction are very important for that. And so we're focused on those interaction. And secondly, the second goal is to use those methods to try to get a better understanding of the molecular mechanisms that are involved in the regulation of the dynamics of glutamate receptors at the synapse. And so in the Nature Chemical Biology article, you can find a description of this approach applied to AMPA receptors, while we've also published a couple of months ago a report on NMDA receptor with a group of Laurent Groc, and that's been published in PNAS. So back to the Nature Chemical Biology. This article is the description of the approach to develop tools that can acutely disrupt the synaptic retention of AMPA receptors. And it can be viewed as the presentation of the genesis of this approach, in the sense that we present the hypothesis that led us to develop those tools. And this hypothesis is that multivalency is an important factor in the stabilization of glutamate and AMPA receptors at the synapse. And so we apply this hypothesis to design biomimetic tools. And by biomimetic, I mean that we're trying to replicate the way we think those interactions are established between the receptors and the scaffolding protein, the PDC domain of the scaffolding proteins. And in the end, we designed divalent ligands that are composed of two PDC domain binding motif derived from the subunits of the AMPA receptors. And so the paper can be divided into three main parts. The first one being the design, where we use structural biology and obviously synthesis. The second one being the selection and characterization of the ligands, and we use for that biophysical methods, freight-based assays in cells, and affinity-based isolation of the cellular targets. And the third part is where we look at the effect of those ligands in neurons. And for that we use single receptor tracking and measurement of glutamate receptors currents. And so the take-home message of this article, of this work, is that first of all, we've been able, by using that biomimetic approach, to generate efficient ligands that can acutely disrupt the synaptic retention of AMPA receptors. Secondly, it would be that by doing so, we've been able to verify that our hypothesis, where multivalency is important, is indeed true, and it does enhance the stabilization of AMPA receptors. And thirdly, and importantly compared to other methods, those tools allow us to work on endogenous receptors with a much better temporal control. And so in the end, and what we come next, so we've validated those tools, and now we can start using them to address specific questions. And in that respect, the article in the PNAS with Laurent Groc on NMDA receptor is a good example of what can be achieved with those tools. And finally, considering the prevalence of clusters of PDC domain at the PSD, we can also start considering implementing that approach to other interaction systems. Thank you for your attention.